Hi, Michelle Glass here, and welcome to our final installment in our Chapter 5, Lecture 5 series. And our topic for this presentation are enzymes. Enzymes are really cool molecules. They are described as catalysts. Catalysts are molecules that are involved in chemical reactions without being changed in any way as being part of the reaction. So they're not broken down, they're not reactants, they're not products, and they're available to continue being a part of multiple reactions because, again, they're not changed from being involved in a reaction. You should think about enzymes as typically being protein, so all those rules about protein structure that we learned back in Chapter 2 would apply. Some of our enzymes are RNA molecules, so we'll see that to some extent, but for the most part, it's fair to think about them as proteins. They're very, very specific. And again, this ties back to that idea of protein structure being so specific. What we will see is each different chemical reaction will have its very own specialized enzyme. And very often, enzymes are named with an ASE ending. So sucrase, or acetylcholinesterase, or catalase. You may not know what each of these do, but you can recognize, because of the ASE ending, that they're enzymes. Now, every name that ends in ASE is an enzyme, but not every enzyme has an ASE ending. So it's not always there. And then for us, we should really consider that in order for a chemical reaction to happen in the cell, it has to have an enzyme. So what that means is that cells can control what chemical reactions they can do based on what enzymes they are making. Now here's an example of a reaction. So we have the molecule sucrose and water being broken down into glucose and fructose. So sucrose is a sugar, it's table sugar that we eat. We need that glucose in our cells in order to make ATP. This reaction will spontaneously happen over a period of years. So you have sugar in your coffee and you're expecting, some of that sugar to help give you the energy that you need for your day, but you have to wait years. Okay, that's pretty ridiculous. So the cell's gonna make a special enzyme. Look at its name, sucrase. So it's named based on the substrate molecule, sucrose, that it binds with. And it's got that ASE ending, so we can recognize that it's an enzyme. Once you stick that sucrase molecule in there, you have that enzyme, and it happens like that. It happens in seconds. So now the cell can use that glucose to make ATP energy. Now, here's an example of what we're talking about. So here in this graph, we are looking at how much energy it takes for those reactants to be converted to products. And this is a very generic diagram. So maybe it's the sucrose and water as our reactants and our glucose and fructose as our products, or maybe it's some other reaction. The amount of energy that it takes is called the energy of activation. And reactants typically have to absorb energy, and then those molecules have to bend in order for those chemical bonds to break, in order for new bonds to be formed. And so that usually takes a lot of energy, and it might take a lot of time, like in that previous example where it took years. With the addition of your enzyme, you drastically lower the amount of energy that is required. So not only are we talking about it's saving time because it's saving the energy that's required. So we call enzymes catalysts, and we talk about them as lowering the energy of activation, or the amount of energy it takes to go from our reactants to our products. Now, how is this working? Basically, that substrate or those reactants will bind into the enzyme, and based on the way they fit with the enzyme, it might be a really awkward kind of bend position, and that allows those chemical bonds to break more easily because they're already kind of contorted or stressed in their folding pattern. Or if we're talking about a synthesis reaction, it might be where you're bringing them in close proximity to each other and making it really easy for that bond to form. So your substrate, your molecule that's binding with your enzyme, can either, kind of be shaped in a funny way to stress those bonds so that they break more easily, or bring them closer together so that bonds form together more easily. So that's essentially what's happening with our enzymes. Now, we've mentioned this word substrate before. Substrate is the molecule that binds to the enzyme. And enzymes and substrates are a very specific match. They are made for each other, right? A lot of times this is described as a lock and key model fit, where the substrate will only fit with this enzyme, nothing else. And so again, a cell can control what chemical reactions can occur by determining what enzymes it's making, because that enzyme is not universal, able to go to any reaction, but just a specific substrate. We can be very specific in what reactions a cell can do. Once that substrate fits into the enzyme, we get what's called the induced fit. So it's like an enzyme substrate hug. And so that enzyme will kind of connect in around that substrate, really helping to lock it into place. So in this case, maybe the way that it's fitting in is stressing out the bond between the different parts of that molecule, making it easier to break. There are lots of different factors that affect enzymatic reactions. There's going to be 3 that we'll talk about. So the first is concentration. The more enzyme that you have produced, the faster the rate of your reaction, because remember, each enzyme has to hug that substrate. So the more enzymes you have, the more free hugs you have for your substrate molecules, so the faster that reaction will occur. Well, the same is true about increasing your amount of substrate. So the more substrate you have, the more can fit in with those enzymes, free hugs, the faster the rate until saturation occurs. So once you have completed all of the reaction, you can't do any more. So there is a limit to how much chemical reaction it can do. The next characteristic or factor affecting enzymatic rates is environment. So here's our second one. This goes back to the fact that enzymes are most often proteins. Proteins have specific shapes. Those shapes determine their function. Those shapes are dependent on environment. So in order for enzymes to have the proper shape, such that they can fit in with their substrate, they have to have an optimal temperature and optimal pH conditions. Now, like for our bodies, that's something that's regulated by our body. So we're maintaining kind of almost the same body temperature and almost the same pH and other factors, in part so that our enzymes will be shaped correctly and work correctly. If you messed up the shape of your protein, that's called denaturing. So temperature and pH can both denature your proteins. Now, sometimes that's permanent, forever change. The shape is messed up, the enzyme is messed up, it never works again. Okay, so sometimes denaturing is permanent. So that's usually like with heat. So you know this about cooking food. When you cook your food, you're denaturing those proteins. So you are changing their texture and their color and their taste because you are denaturing those proteins. And you know you can't take it back to a raw food after you've cooked it, okay? But denaturing is not always permanent. Sometimes you can denature and then renature, refold that protein, or in this case, enzyme. And you'll see that more with pH. But pH has to be regulated so that your enzymes will be able to work correctly. The third category we see is the presence of what are called cofactors. And these are little helpers that are made in the cell. You have different types. You have some that are inorganic cofactors, and then you have what are called organic coenzymes. Both of these are functioning as helpers. Those organic coenzymes, when we talk about them with our diet, we're thinking about vitamins as examples here. The last thing to look at is how the cell can regulate enzymes. So there's 2 different ways. One way to regulate an enzyme is by making what's called a competitive inhibitor. So the competitive inhibitor is fitting into the enzyme in such a way that the substrate cannot fit in. So it's blocking what's called the active site. If that competitive inhibitor is in the way, the substrate can't bind and the chemical reaction will not happen. So cells can regulate the functioning of enzymes by making competitive inhibitors. Another type of regulation is what's called the non-competitive inhibitor. In this case, what happens is that non-competitive inhibitor binds somewhere else on the enzyme, changing the shape of the enzyme so that it no longer fits with the substrate. So in each case, you're preventing the enzyme and substrate from binding together, but you're doing that with different mechanisms of action. And again, this is another way that cells can regulate which enzymes are functioning and which are not. You could take that non-competitive inhibitor off, and now the enzyme is working again. Okay, and then we are able to manipulate this in our design of drugs and pesticides and poisons. A lot of these are acting as enzyme inhibitors, and again, by changing which enzymes are available, we are changing the chemical reactions that are happening in a cell. And that completes us on chapter 5.